Hi, it's my great honor to give a company presentation today, China Healthcare Summit today. I'm Yu-Kyeong Hwang of LabCell, Head of R&D, Senior Vice President. I'd like to give a presentation of a company overview and then move on to our R&D program, including the NK cells and the stem cell program. Okay, I'd like to start with our company profile presentations. GC랩셀 is one of the subsidiary of Green Cross family. It's established in 2011 and listed in the Kosdaq market in 2016. We have two subsidiaries, shareholder subsidiaries in Korea and America. One is GCL and established in August 2019 and supported each page of the new drug development from consulting service to the clinical trial report. Another one is Artiva, established in March 2019, is focusing on the development and the commercialization of NK cell program in USA. As I said before, we are one of the subsidiary under the Green Cross Holdings. One of the cell and gene therapy company among 28 subsidiaries. We are focusing on the cell and gene therapy program in Korea and the worldwide program too. Based on our cash cow business, such as reference laboratory service and related central lab business, we have enough cash cow business. And then we focusing on to the gene and cell therapy program as a new growth engine project. Now, I'd like to move on to the NK cell program here. We have 5 categories of NK cell program, from early discovery to late stage program. We are focusing on to the manufacturing first because NK cell must be supplied huge amount of cells as a drug. So first we are focusing on to the manufacturing and the freezing process through the feeder cell systems and bioreactor systems. Finally, we can produce the bunch of the NK cells from peripheral blood and cord blood too. This first program is done in phase 2A in Korea for hepatocellular carcinoma. And then we think about higher efficiency using the antibody combination. Compared to the T cell, NK cell bind antibody by its natural property like CD16 receptors. So when it combined with the antibody, any kind of antibody, we can improve their functions. So we finished the phase 1 in Korea against the recurrent refractory lymphoma patients. And next, like CAR T cell, we would like to engineer the CAR NK cells against many kind of solid tumor and hepatocellular carcinoma tumors. That kind of solid tumor program will start preclinical study in next year. And then, as like gene editing technology using the CRISPR-Cas9, we can make gene-edited NK cell too. Finally, we finalized the candidate, so we shortly move into the development stage. Next, the most early stage program is iPS-derived NK cells. For this all kind of program, we have multiple partners globally. I'd like to explain our first peripheral blood NK cell program. The first clinical trial is done in phase 2 against the liver cancer. We enrolled 78 patients for 2 cohorts. And another one is antibody combinations against the lymphoma patients. As you can see here, we can see a very nice synergistic effect in animal study. So we entered the clinical trials phase 1, and then finally we got some data and report in the global conference EHA last June. And also we can present our result in the ASH this December. Next, we are focusing on to the cord blood-derived NK cells. Compared to the peripheral-derived NK cells, actually cord blood shows very low donor variations. So, if we can use this cord blood, then we can provide a very unique, very homogeneous product for multiple patients. However, cord blood has some limitations. The NK cell in cord blood is not activated very easily, so we produced our own feeder systems and the bioreactor systems too. Finally, from the one cord blood unit, we can provide a couple of 10,000 product from single cord blood. As you can see here, we can see a very nice efficacy in vitro and in vivo. So this program is licensed out in the Artiva in US company, and the program is submitted to US IND in last August. And also we are developing the CAR-NK project. CAR-NK program is very important to increase the functions and the efficiencies. However, the issue is in the intracellular domain. So we screened our own intracellular domains, NK specific, so that is issued by IP. So this program is entered clinical trials next year. As like other gene-edited programs, we also delete some unnecessary genes like immunogenicity induced gene or some inhibitory genes. If we remove the inhibitory genes, then NK cells survived and persistence in a body for long times compared to the convenient NK cells. Even when it delete inhibitory genes, we can see very nice efficiency in a tumor microenvironment too. The final program is against for COVID-19. Actually, NK cell kill the tumor and virus-infected cells through their natural property. So we can use these NK cells against the COVID-19 patients. The safety is already proven, so we can easily use these NK cells for the clinical trials. And also we have partners who is developing the antibody recruiting molecules. Then this antibody recruiting molecule arm is bind to the COVID-19 and then recruit immunoglobulin by its nature. Then this kind of mechanism works as a virus neutralizing agent. Therefore, we have two different programs. One is NK alone for COVID-19. Another one is arm combinations for COVID-19 too. This program will enter the clinical trial shortly. Here is a kind of summary of our GC랩셀 technology. So we already proven the safety even in the non-related donor-derived expanded NK cells. And also, we can achieve the mass productions and the cryopreservation technology, so we can guarantee 2-year survival and stable data for NK cells when it preserved in the liquid nitrogen tank. We also have our own IP in CAR constructions and gene editing programs. And all of these NK cells are used by international standard cord blood from New York Blood Bank. It is proven by FDA license, so there are no problems to use the NK cells. Here is kind of chart compared to our competitors. As you can see here, we have a very multi-layer of technology using the NK cells and the manufacturing, freezing, And also we have a multi kind of programs like CAR-NK, NK alone, or memory-like NK cell, etc. So I think we have very strong powers to use our NK cells with Artiva biotherapeutics. Here is our our collaborators in domestic and worldwide. We have multiple antibody partners like Yu-Kyeong in Korea and MorphoSys in Germany to combinations with antibody treatment. And also we have Ph.D. Therapeutics in Canada to use their shuttle systems in delivery of the specific gene and proteins. Artiva, as I said before, they will develop our NK program in globally, and Universal Cells in US for iPS collaborations, and Cleo for ARM collaborations too. Now I'd like to move on to the stem cell program. Actually, we have a very special source of the stem cell. It comes from tonsil. Tonsil is a very unique organ, so tonsil-derived mesenchymal stem cell has very strong anti-inflammatory powers, and also it is very strong proliferation power. So we give another stimulation in vitro, then we can get a very excellent anti-inflammatory functional mesenchymal stem cell. So the first program is focusing on to the psoriasis. As you can see here, psoriasis is a very complicated immune disease. However, we evaluate their functions, mesenchymal stem cells function, by blocking with IL-23 and IL-17, or even in the interferon gamma. So hyperactivated immune system can be controlled by tonsil-derived mesenchymal stem cells. This psoriasis program will enter the IND submission next month, so we expect dosing the next year too. Of course, this anti-inflammatory function of a mesenchymal stem cell can work for COVID-19 too. In the COVID-19 case, the severe COVID-19 patients received very strong immune activations in their lungs. So when it injected the tonsil-derived mesenchymal stem cells, then this cell can inhibit hyperactivated immune system. So we expect the IND submission very shortly with Cleo and other partners too. Here are the summary of our pipeline. I would like to emphasize our NK program first. We have peripheral-derived NK cells. The most advanced program is done in phase 2, and another antibody combination phase 1, and the CAR-NK program and engineered NK program will start preclinical or sometimes in the clinical study in global stage. And also we have a stem cell program against the psoriasis and the COVID-19 too. We are looking for the partners for NK program and stem cell program, especially NK cell program. You are looking for the strategic partners and the out-licensing partners in collaborations with CAR program and the PBNK, CBNK too. For the stem cell program, we are looking for a new technology and for in-licensing. We would like to improve our stem cell program, so we are very happy if you have a very special and unique technology for stem cell area, then we would like to meet very shortly. And also we have two kinds of COVID-19 project, NK cells and stem cells, so we are very happy to look at partners for clinical development in China. If you have any questions, please contact these two guys, Isaac Lee and Victoria Hwang. Thank you for your time. I'm very happy to see here. Nice to meet you. Thank you.